Good morning and welcome to the 2018 Georgie Palade Award and Distinguished Lecture. I'm Larry Grossman, Director of the Center for Molecular Medicine and Genetics here at Wayne State. And it's my great pleasure to open this ceremony, followed by the distinguished lecture by this year's awardee, Professor David Baltimore. I'm also delighted to inform you that there are two anniversaries here. One is the 15th anniversary of the Palade Awards, and it's the 20th anniversary of the founding of the Center for Molecular Medicine and Genetics. The center was designed in an interdisciplinary way, comprising basic researchers, physician scientists, computational biologists, and genetic counselors. And this interdisciplinary mix has allowed us to carry out a great range of activities, all designed to both unearth basic knowledge and to improve human health. We've even had several startups. We also have tried not to lose sight of the fact that we should have fun doing research. The Palade Award was established in 2003 in recognition of the lifelong contributions of Professor George Palade, considered the father of cell biology, who discovered various cellular structures, including the ribosome, for which he received the 1974 Nobel Prize in Medicine and Physiology. Since the inception of this prize, 14 awards have been made, among them to 5 Nobel laureates, 3 of whom actually received their Nobel Prizes after receiving this award. This year's ceremony is made special in another way, and that is we have with us 2 previous recipients of the Palade Award. One is Professor Banu Jaina, who is a member of the Wayne State faculty. He's Distinguished University Professor of Physiology and a member of the Center for Molecular Medicine and Genetics, who received it for the porosome, the universal secretory portal in cells, and also Professor Walter Boron of Case Western Reserve University, who is right up front. Stand if you would like, so people can see you. Thank you for making the trip. And he won for basic discovery of pH regulation in cells. The discoveries were made by both when they were in their 30s and were colleagues at the Yale University School of Medicine. So it's now my pleasure to invite Dr. Jaina to present this year's award. Professor Jaina.
Good morning and welcome to this wonderful ceremony and presenting this award to one of the most famous biologists that have lived in our times. And this is also a special occasion for me because it's a great honor to be selected to present this award, and it's quite humbling actually, because not only as a graduate student, but even as a high schooler, I've read some of his work in our biology textbooks, so it's truly humbling. Professor David Baltimore, for this award is an outstanding individual, both as a scientist, as an educator, administrator, and a public advocate for science and education. And actually, he needs no introduction. But I will speak a few lines on his lifelong contributions that continues to this day. Professor Baltimore is considered, as I said earlier, one of the world's most influential biologists whose contributions continue to have profound global impact on science and education and society. Among the numerous awards and honors he has received for his scientific contribution is the 1975 Nobel Prize in Physiology or Medicine at the young age of 37 for the discovery he made when he was only 32 years old. And just to remind you that most of us receive our NIH grants at the average age of 44, just to have a comparison. He is the founder director of the famous Whitehead Institute, established in 1982, where the young mathematician turned biologist, Professor Eric Lander, who has visited this campus before, led the successful sequencing of the Human Genome Project from '86 to 2004. The Whitehead has become a Pathfinder Institute worldwide, and to many subsequent research institutes, including the famous Broad Institute, established by Professor Eric Lander. Professor Baltimore has served as president of both the Rockefeller University and California Institute of Technology, and has led numerous national and international committees on science and education for the benefit of society. Professor Baltimore has played a major role in the development of biotechnology in the United States and in the world since the 1970s. He has helped found many companies such as Calimmune, Immune Design, S2A Molecular Inc. He has also served or serves in the board of directors at several companies and nonprofit institutions, including the Broad Foundation, the Broad Institute, Amgen, Regulus Therapeutics. He's a member of numerous scientific advisory boards, including the Broad Institute, the Rogan Institute, which was recently developed a few years ago, and the Column Group, and several other institutions. He's the director. He directed the Swiss investment firm BB Biotech, and he has authored more than 700 research publications. And I happened to just screen through some of this year's publications. Beautiful work that he has done or continues to do. His lab produced about 8 or 12 papers this year. Professor Baltimore, congratulations on receiving the Georgie Palade Award and gold medal for your pioneering discovery of another new important area, primarily the transcriptional factor NF-kappa B, and for your many contributions to the field of immunology. Professor Baltimore. What I would like to do now is request Professor Wall, our Associate Dean for Research, to conduct this lecture. Professor Wall.
Thank you, Bono. First of all, I'd like to welcome everyone to this event. It's truly an honor for me to be able to introduce Professor Baltimore to present his lecture and remind the audience that Dr. Baltimore will be happy to take questions after he finishes his talk. He's going to be speaking with you today on his most recent studies, which are the fine control of gene expression by retaining introns. Professor Baltimore, the floor is yours.
Let me change this.
It's on? It's on.
It's on. And this you can,
Yep.
or you can, it's next.
Okay. Excuse me?
You're going to be the driver.
Yeah, I'll take care of it. There we are. Good morning. It's a pleasure to be here. It's an honor to be associated with George Palade in this way. George was a man I knew quite well because I received my PhD at Rockefeller University, where George was at that time, running a huge laboratory in cell biology. And I learned from him the beginnings of cell biology, an area that has just flourished in the intervening years. So he's a man I have the deepest respect for. He also, as an individual, he was a man of great humanistic understanding and education, sort of a Middle European intellectual, as well as a great scientist. He was a man who acted as an advisor to many institutions in American science, and he would turn up in the oddest places because somebody wanted to get expert advice, and George was so generous with his time and his knowledge. The topic I've chosen to talk about is very different from things that I've done previously in my scientific career. It's work that we have fallen into, and I'll explain sort of how we got there. But it reflects a surprise. And the reason why I'm going to talk about it is because I think many of you will find it surprising. We all know about the splicing out of introns. If anybody doesn't know about the splicing out of introns, you're in for a dose of difficult science. And we know that messenger RNAs, mammalian messenger RNAs, are made as very long precursors, and then regions are cut out, the introns are cut out from them. And at least I always assumed that that was a mechanical process, that it just sort of happened. And I never run into the fact that it's a controlled process and that certain pre-messenger RNAs splice very easily and other ones with much more difficulty. But I'll show you that that's true. And although I can't answer all the questions that are raised by the observations I'll show you, it's pretty clear that it represents a type of control mechanism that was not previously understood. So it all comes about because I became interested in inflammation. And I became interested in inflammation because, as was mentioned in the introduction, we discovered the NF-kappa B transcription factor in my laboratory in 1986. And it turned out, and that's not why we had discovered it, that it is the controlling factor for the inflammatory response. So one of the most significant changes that can occur in the body is activation of an inflammatory response. And in cases where that is a massive activation, it's life-threatening. In general, in a normal inflammatory response, like for instance, if you get a cut and it becomes inflamed, it becomes red, the process occurs in waves. First, neutrophils come into the region. They secrete inflammatory cytokines. That's part of how the whole process evolves. Macrophages come into the area. They start chewing up the bacteria that have been killed by the neutrophils, as well as the neutrophils themselves. Lymphocytes come into the area and act both to make antibodies and to provide T-cell responses. And so those are all of the blood elements that come to an inflamed area and act to counter the infecting organisms. And then there's a cleanup process. And the cleanup process is a somewhat extended process, which involves proteases that chew up the garbage that's lying around, and then everything calms down. And NF-kappa B is at the center of all of that. And so it's maintaining a control over processes over long periods of time, and long can be days. And we don't usually think of transcription factors as having carrying out a process of control over long periods of time. I tend to think of transcription, at least I think of transcription factors, as activating transcription of a particular gene at a particular moment in time. And if you think about the Drosophila transcription factors, they act sequentially. There's not one transcription factor, but many of them that activate sequentially and lead to the sequential steps of Drosophila development. So NF-kappa B is actually in all cells. It sits there quietly, not doing anything, because it's held in check by an inhibitor protein called I-kappa B. And all of the different ways that inflammation can be activated, and there are many of them, cause the degradation of I-kappa B, the release of NF-kappa B, and that released NF-kappa B moves to the nucleus of the cell and activates many genes for transcription. And many means hundreds of genes. And there are lots of other ancillary proteins that are activated, the inflammasome. I won't go into all of those. But the central role of NF-kappa B makes it a particularly interesting subject to understand, and we have been trying to understand it for a number of decades. Another way of looking at the activity of NF-kappa B is in this schematic diagram. Let me see. Does this have a... No, oh, it doesn't have a pointer. But I just managed to turn it all off. Got it back. I can, what, there's a... Oh, I see. Okay. Right. So, yeah, I'll go back. Maybe. So, NF-kappa B is latent in cells, and many, many different stimuli, bacterial lipopolysaccharide, tumor necrosis factor, IL-1, can all activate it out of latency. They all do the same thing. They cause I-kappa B to be degraded. That induces NF-kappa B activity because it now can move to the nucleus. You get the response, and then you get a resolution of that response. And this, as I said, may take hours or days, but it has to happen. And then it puts the system back in latency. If you don't get resolution of that response, if there's continual activation, for instance, then you get a variety of pathologies. And those pathologies are manifold and affect many parts of the body. But the things that are most interesting, at least to me, are its role in cancer, where chronic activation of NF-kappa B leads to cancers, and the general area of chronic inflammation, which is today thought to play a role in heart disease and other ills of the body. And so we're talking about one of the major sources of pathology. So this is another way of looking at the activation of NF-kappa B. Here it is. It's a dimer. It's bound to I-kappa B. And all of these different activators can activate the degradation of I-kappa B, the release of NF-kappa B, and its movement to control hundreds of genes in the nucleus. And it does that by activating something called the IKK complex, which involves phosphorylations, and I won't go into those details, but you can sense from the number of activators that this is a pleiotropic system of great importance. So how do you manage to have one transcription factor activate hundreds of genes? You could have in all those 100 genes some kind of stereotypic response element, and the same thing could happen at all the genes. But that's not true. In fact, each gene has its own response elements of the hundreds of genes that are involved. And actually, NF-kappa B is not a single transcription factor. It's a small family of transcription factors that come together as hetero and homodimers from a pool of 5 monomers. It's also subjected to secondary modifications, phosphorylations, glycosylations. There is regulation of NF-kappa B by trans regulation through non-coding RNAs, particularly microRNAs. That's a whole separate discussion that I won't get into. But the important point, and the one that will drive the rest of this discussion, is that the genes that are induced with their own particular time courses. Some are induced very rapidly, some are induced slowly, and as you might think, the ones that involve the immediate response, for instance, the neutrophil response, are activated very rapidly. The ones that involve, for instance, cleaning up after an inflammatory response are induced much more slowly. So I want to focus on that, on why some genes turn on very quickly and some genes turn on slowly. It's an ongoing process of investigation, and we don't have all the answers, but we have some interesting answers. And it all comes about because of this observation made by a postdoc of mine, Shengli Hao, now a decade ago. And the observation is this, that if you induce a cell type, and it could be a fibroblast, it could be a macrophage, with an inducing agent like lipopolysaccharide, or in this case, tumor necrosis factor, and measure the turn-on of genes, within half an hour after treatment with the inducer, there's a collection of genes that turn on, and it's these red genes here. And if you go out 2 hours, there's now many new genes that turn on. And if you go out 12 hours, there are many more new genes turned on. And in fact, a lot of the original genes that were turned on are turned off. And so, because of the way the experiment's done, I can group them as group 1, group 2, group 3, and we'll refer to them that way. But they're not precise groupings. They have overlaps. So group 1 genes are turned on with a peak at about half an hour, group 2 genes with a peak at 2 hours, and group 3 genes peak very late. And there are lots of them. There are group 2 genes, group 1 genes, there are 25 of them. There are 57 group 2 and 100 group 3 genes. And those numbers aren't even, are underestimates. So here are a bunch of group 1 genes. You can see that they turn on very rapidly. They come to a peak. They fall. And this is just measuring messenger RNA of these genes. Or it could be measuring actually proteins too. But the messenger RNAs are particularly unstable, and so they turn on quickly and then are turned off quickly by their instability. Group 2 genes turn on and remain on for some time. Group 3 genes take a long time to get going, a couple of hours, and then accumulate over time. They're actually rock stable and accumulate for hours, if not days. And so we have 3 different patterns of the group 1, group 2, and group 3 genes. And now we come to the question of what is controlling that pattern. So what the best guess, the one that I must say I would have made, is that the group 1 genes are just intrinsically unstable. They turn on and turn off. That the group 2 genes probably have multiple transcription factors that have to work on them, and so they take a while to get going, and group 3 genes have many more transcription factors to get going. And so what you're looking at is 3 different times of turning on genes. That turns out not to be true. Wait a minute, I'm going to, yes, that turns out not to be true. So you turn on group 1 genes, you turn on group 2 genes slowly and group 3 genes very slowly. But if you measure not messenger RNA, but pre-messenger RNA, that is the unspliced transcript from the gene, then they all turn on at the same time. And it's about 5 minutes. There's about a 5-minute lag after induction, and each of them turns on over the next minute or two activates transcription. So it's not a graded transcription process. It's a graded something else. And that then became the question, what is going on here? So we conclude from that work that Sheng Li did that all genes induce transcription coordinately, roughly, that the group 2 and group 3 genes lag in some kind of processing event, And then there's another aspect to NF-kappa B-induced transcription that any of you who've worked with it will be aware of, and that is that it's oscillatory, and so there's a cycle of induction and repression that has about an hour period, and I'm not going to talk about that at all. I'm going to pretend it doesn't happen, but it's very interesting in and of itself. So, if it's not due to the sequential initiation of transcription, but to some post-initiation event, what's the most likely answer? And the most likely answer is that there's some kind of control of RNA splicing. And in fact, we looked at that question directly, and that turns out to be true. That's not what this says here. Sheng Li had understood that, and we published that some years ago. And about 5 years ago, a couple of other people in the laboratory have taken up this problem, and they decided to apply a very powerful technology, which is RNA sequencing. And the problem then is, can you sequence the newly made transcripts after induction? Well, they're made so massively that you can, in fact, see those transcripts against the background of ordinary transcription, and they really stand out. So we then set about to try to purify those transcripts and characterize their behavior. And what we wanted to do was to be able to see the unspliced RNA being made and then see it being spliced sequentially over time. And to do that, we had to select out the induced pre-messenger RNAs separate from the background of other transcription that's going on in the cell, because there's lots of other genes, housekeeping genes, that are being transcribed. And work of Steve Smale and Doug Black at UCLA showed us where to find these RNAs. And the answer was in chromatin. What they had shown was that if you induce an inflammatory response, the first place you see newly made RNAs before they are completely spliced is in chromatin. It's attached to the chromosomes. And we don't exactly know what's holding them there. We don't know what's holding them there. And we do know that once they're completely spliced, they're released from there. But we don't know what's going on in the intermediate stages. All right, so the two guys who took over this problem are Dev Majumdar and Luke Franco. And they developed this methodology for finding the newly made transcripts. They started by treating macrophages with tumor necrosis factor. That's a good pro-inflammatory stimulus, and macrophages make a very big response to that. We isolate the chromatin-associated RNA, knowing that that's where the gold is. But there's a lot of chromatin-associated RNA which we don't want to purify, and so we fractionate it, and we fractionate it by making cDNA copies of all of the RNA. That's pretty easy. You can prime with oligo-DT. They all have poly A at their 3' end. And so, in fact, we'll only look at completed transcripts. Transcripts that are in the process of being made would not be picked up by this method because they wouldn't have poly A on them. Think about it. And then we enrich for the transcripts that we want by hybridizing these cDNAs to oligonucleotides of complementary sequence to the last exon of the RNAs. That guarantees that we only get, again, full-length transcripts. It's another form of purification. And we use biotinylated probes for that so that we can capture them with streptavidin. And so here we can see that some of the transcripts will be captured by the complementary oligonucleotides, and others won't, and that we can bring down the ones that we want with streptavidin, with immobilized streptavidin. So now we have the transcripts we want, and what we do is sequence them. And we use Illumina sequencing, so we sequence little pieces of them and then reconstruct the RNAs. And this is a method that I should give credit to Mitch Gutman's lab. He's a professor at Caltech. And what we do then is to look at all of the individual transcripts. So right in this particular slide, we're looking at the transcripts for the I-kappa-B alpha gene. It's an induced gene, induced to high level, easily seen in macrophages. And there are two different ways of showing the same data here. So this is the panel on your left is over here, and the panel on your right is over here. Let's just look at the left-hand panel for the moment. This is a linear plot of the transcripts that we find in the precipitated RNA. And what you can see is that there's lots of RNA that's near the 3' end, and there's some RNA that goes all the way down to the end, and it's mapped here against the gene structure that's known for the I-kappa-B alpha gene from DNA sequencing. And the thick bars are the exons, and the thin bars are the introns. And what you can see is that there's no transcripts accumulated that have intron structure. The only thing that's accumulated is exon structure. So what that says, and it's an important conclusion of its own, is that in these long transcripts, which have been picked up, remember, by their 3' sequence, that you have completely spliced out all of the nucleotides that represent the introns. And that's true of the first intron, it's true of the second intron, it's sort of true of the third intron, and by the time we get to the fourth or fifth intron, there is some sequence here because it hasn't been completely spliced out. And so that shows you what goes on in the manufacture of a messenger RNA, that as the polymerase goes by, the splicing factors get onto the RNA and splice out, and you can calculate that that happens within about 20 seconds of the polymerase passing the border of the intron. So splicing takes about 20 seconds on nascent chains that are attached to chromatin by something at their 3' end. Now, I said these are linear plots. So if you look over here at 0 time, you can't see anything. And if you look at 4 minutes or 6 minutes, you can't see much. And by 8 minutes, you begin to see the transcripts. But it's not that there isn't anything there. It's that by using a linear form of analysis, there is so little there that you can't see it. Actually, you can see just a hair. So what we did is transform all of this data into a logarithmic form, and we scale that logarithmic form by the maximal point, which generally turns out to be very near the 3' end. So if we look at 0 time, now we can see the 0 time transcripts. And they have the same characteristics. They're spliced in their first intron, spliced in their second, and that's actually uninduced RNA because it's at 0 time. And all of the rest of them are different from the 0 time by having transcripts evident in the introns. So they're not completely spliced. And we've caught them in the middle of splicing. And it depends on the length. If it's very long, you wouldn't see this. If it's short, you will see this. Now that's very powerful because that gives us a chance to begin to ask the questions we want to ask. And it's all because of using logarithmic analysis, so that the amount of transcript actually here, for instance, is only a couple % of the maximum, but you can see it perfectly easily because of using a log scale. So now you can understand how to look at the data that we will be showing you. And we used 230 different transcripts. So we have the data from 230 transcripts like I kappa B, but of all sorts of different functional importance. And it's a great opportunity to mine all of that data. But we want to know how to quantitate whether splicing has occurred or not. And so we use what's called a co-SI, coefficient of splicing an intron. And it involves calculating how many of the reads that are made on the Illumina sequencing show complete splicing out of an intron, as opposed to ones that don't show complete splicing out, or don't show any splicing out of the intron. And I won't go into the mathematics of it, but it provides a very nice quantitation. So instead of having to look at this, we can now look at a number that characterizes the intron. And we can plot that number over time. So here's 50 minutes of data. And let's just focus on the 5th intron. And what you can see is at 0 time, that is the uninduced RNA, has a fair amount a fair coefficient of splicing of around 0.8. And that as you induce it, that goes down to 0.5 and down to 0.3 because we've induced a lot of new RNA, and that RNA has not entered the splicing machinery, or the machinery hasn't entered the RNA. But over time, it begins to, and so it goes down to a minimum and comes back up. And that's a very characteristic, and it's characteristic of introns near the polyadenylation site. But the ones down here are just stay up at the top and have a coefficient of 1. Because they are so far from the place where we're capturing it that they will have spliced 100%. And the ones in between are obviously in between. All right, so this is what one gene looks like, and this is what all the genes look like. And what that tells you is something very interesting, which is that each gene has its own pattern. It's not that there's some canonical pattern that all genes behave like. Some of them are very slow to splice, some of them are rapid to splice. And the best way to show this is with what's called a whisker plot, in which we have the number of transcripts that are way down here shown by individual dots. So that's one transcript, that's another transcript. And then there's a straight line, which is things that are much more populated, tenfold roughly, excuse me if I remember correctly. And then there are ones that are even more populated, and this black line up here is a median position. So I'll just show you whisker plots. And what you really want to know is, why are there some dots down here after 50 minutes of induction that have not spliced? These are co-SIs that are very low. And so we pull one of those out and take a look at it. It's from a gene called CXCL10, but obviously we could look at all of these 230 genes. And what you see is that even though this intron is in the middle of this transcript, there's a lot of it unspliced. Much more than there is in the intron that's further 3'. So this intron is being held up from splicing and isn't acting as it should, or as we might have thought it would, from its position in the messenger RNA. And in fact, it goes on for a long time. So even at after 50 minutes of time has gone by, it's still sitting there. And we call these bottleneck introns. Here's one in the CD40 gene, here's one in the DAX gene, here's one in IRF7, which we will come back to later. It's in a lot of genes. They're all genes of class 2 and class 3. No gene of class 1 has such an intron in it. So we're beginning to now understand a little bit why this process is distributed in time. there are lots of genes being held up from splicing. Now, this is to remind you that what you should have learned in elementary molecular biology, that introns have a 5' splice site, which is highly conserved. And it has 2 G residues that take up 2 positions, a T residue, so you have a GT here, and other conservations. And this gives you a consensus. And so most introns have a structure close to the consensus at the 5' end. But one of these slow to splice introns, if we look at the sequence around the 5' end, we'll see that it's not canonical. So here's the IL-27 gene, just a gene which is conveniently illustrative. Intron 2 is GTGAG, which is a canonical sequence. Intron 3 is GTGAG. Intron 4 is GTAAG, which is an even more canonical sequence. And intron 4 is GTAGA, which is a poor sequence. So here's the reason why this intron is not splicing well. It doesn't have the canonical sequence. It has a variant. And it's a bad variant. And you can say, well, maybe that's just because it's mutated in this species. This is the mouse. And if we look at it in the rat or human, it'll be canonical. No, sir. If you look at it in the mouse, the rat, the gorilla, and all of these species, it's always the same and always non-canonical. That says that evolution has maintained this non-canonical sequence over the evolutionary span represented by the animals that are here, but we can even look more widely and it's still maintained. So we've learned a lot from that. Now, why is the 5' splice site conserved? It's because the 5' splice site interacts with the U1 SNRP, an RNA, and it has to maintain exactly the right sequence. And if the sequence is off, you won't get good hybridization of the U1 SNRP to the message. And we can actually make that into a parameter, which is almost perfect. Canonical gives you a 10 score, and really terrible gives you a score at 1 or less. And what you can see is that, as I said before, the immediate early genes, the class 1 genes, are mostly up around 10, but the class 2, 3, and it's actually split slightly differently here, include lots that are down around 5 or 3 even. So we learn from this that, first of all, most of the splicing is co-transcriptional. It's occurring while the transcript is being elongated. That there is significant heterogeneity in the rates of splicing. That there are bottlenecks that are really holding up splicing at a particular point. Those are all found internally in messenger RNAs, not at either end. And generally, there's only one bottleneck per transcript. And so we've now learned that what's controlling the response to these inducers is things that are built into the sequence of transcripts. And that in the sequence of transcripts, there's a lot more information than we might have originally thought. That the half-life of a message is found in that sequence, as well as the splicing efficiency and timing. So Luke Franco decided to ask, is there anything special bound to these transcripts, which could be a protein that plays a role in this splicing? And he found such a protein called BUD13. And I don't want to go on too long here, so I'll try to do this reasonably rapidly. In fact, so the gene that I want to look at is IRF7, and it has this bottleneck intron, which is very obvious and splices really poorly. And it doesn't matter whether we induce with TNF alpha or we induce with poly-IC, which is an interferon inducer, or with CPG or with interferon itself, it's held up in all of those cases. And it has a weak splice donor site, a weak 5' splice site. It's also very short compared to other transcripts and very high GC. And those, as you'll see, are characteristics of these bottleneck introns. So here, for instance, looking at this intron, it's what we call the maximum entropy score, which is the measure of the quality of the 5' splice site, is lower than consensus. The intron size is smaller than average, and the GC content is higher than average. So he wanted to see whether there was anything bound to these odd transcripts. So he prepared the transcripts, the RNAs, and he prepared them from cells that had been UV treated so that you cross-link proteins to the RNAs. And I won't go into the technology, but it's actually a beautiful technology called RAP-MS, developed by Mitch. And what he found was that attached to the IRF7 messenger RNA, or the IRF7 transcript, actually, this should read, is a protein called BUD13. And if you look at a whole lot of other proteins of related function, they're not particularly tightly associated. BUD13 absolutely stands out. And again, it doesn't depend on how you induce it. You can induce with TNF, you can induce with poly-IC. And BUD13 is there, attached. So we knocked out BUD13. That's now today trivial. You make a short hairpin RNA and treat cells with it. And what happens is you get a much higher amount of retained intron. Compared to wild type, to not treated. And that says that what BUD13 does is to help these poor introns splice better. Because if you knock it out, they splice more poorly. And we can quantitate that with a delta SR function that I'll leave go of. Now, what does IRF7 do? IRF7 is involved in the interferon response. It actually is a positive feedback regulator of interferon induction. I'm not going to go into this slide, which is a little confusing. But what we found was that if you treat cells with an shRNA against BUD13, and you just look at the amount of the IRF7 transcript, there is a lot less IRF7 transcript that is accumulated over time. And if you look at the amount of interferon transcript, there's a lot less interferon alpha and a lot less interferon beta. So all of this says that you need BUD13 to give you a maximum interferon response. And if you don't have it, you don't get it. And this is the same sort of thing. And so we decided to test this notion by infecting macrophages with a virus, which is interferon sensitive. And that's vesicular stomatitis virus. And what you find is that you make a lot more vesicular stomatitis virus product if you treat cells with the shRNA against BUD13. So BUD13, by reducing interferon, increases the effectiveness of a virus, or the ability of a virus to grow in the face of what would ordinarily be an effective interferon response. So this mechanism, which we've found of BUD13 overcoming the bottleneck introns, has important biological consequences. Finally, is it particular to IRF7? No, not at all. And here are a bunch of transcripts, all of which are affected by BUD13. They're mostly genes that you may never have heard of, certainly mostly genes I had never heard of. But they all gave the same response. Here's the IMP4 gene, whatever it does. It actually splices pretty well without with BUD13. Without BUD13, it accumulates a lot of transcripts. And the same is true of PSMA3, and the same is true of this other one, all of these along here. And they are all small, relatively small introns, and they're all high GC introns. So there's a pattern here. Not that we understand the meaning of that pattern. And they, in general, have poor splicing ratios. And in fact, the poorer the splicing ratio, the more the effect of BUD13. So here's what's actually going on. Here's a pre-messenger RNA. It splices out many of its introns. That's shown as one intron here. But one of its introns splices out very poorly, and so here it is hanging around in a pool in the nucleus. What happens to that pool? Well, it could be delayed splicing. And we're actually trying to figure out how you measure that. It's not easy. But we think it occurs. But it also could be, and that will then go out to the cytoplasm and be ordinary messenger RNA. Or it could be degraded. And there's degradation in the nucleus by the nuclear exosome, which looks for transcripts that have introns in them and degrades them. So it's certainly going to reduce the effectiveness of the transcription of a gene. And if any of it gets out to the cytoplasm, and we actually can't see any getting out to the cytoplasm, but if it does, it will be degraded by what's called NMD decay, another ribonuclease that chews up transcripts in the cytoplasm that are not making protein. And this is only the 5' splice site. So if you look at the 3' splice site and do the same kind of analysis, it's actually all within noise. And is not being read by these mechanisms. And this actually repeats that. So the conclusions that we draw from this work is that IRF7 has this poorly spliced intron that is small, GC-rich, and has a non-consensus donor sequence. BUD13 is highly enriched on these transcripts. Someplace it's binding, we don't know as yet where. And the knockdown of BUD13 leads to increased retention of these poor introns, of the messages with poor introns, and therefore less messenger RNA. And so what BUD13 really is doing is improving the splicing efficiency of these short GC-rich introns with weak 5' splice sites. And it means that evolution, because actually BUD13 is in yeast, it's in higher cells, and so clearly it has evolved to help the splicing of these poor introns. The question that remains is why those poor introns are there in the first place. Because you would think that, that evolution would have just gotten rid of them, and you don't need a whole mechanism to deal with them. And we are wrestling with that question as I speak. This is work that was initiated by Cheng-Li Hao, carried forward by Dev and Luke. Mitch Gutman at Caltech has been a tremendous help in methodology. And with that, I thank you for your attention.
Questions for Scott?
Yeah, Dr. Baltimore, wonderful talk.
Thank you.
A lot of my concerns are really, 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 especially in light of evolution. We, in CMMG, have a history, I only remember the National Academy was an evolutionary genetic Morris Goodman. Have you ever looked at the age, that in the 1918, the age for the Denisovans or the Neanderthals at those
No, we have not looked at other species, particularly the precursor humanoid species, but it would be interesting.
I mean, it's a thing of being human, it's still an
Yeah, but it's a little hard to get a hold of macrophages from denosophages. The sequence would be interesting, yes. I'm sure they're there because they're there in so much more primitive species.
I don't know, but my question is, I'm sure after this talk, you start to think about therapy. Have you identified the gene of interest and tried to create these splicing process to create a protein that can function?
No, we have not tried to bring this to therapy. It would be interesting to try to do that, but we've actually been busy enough on the mechanistic side. You know, it's an interesting question, though. It's an interesting question that you ask, because if this were 20 years ago, or 30 years ago, which times that I can remember very well, I don't think you or anybody would have asked that question. And the reason is because we weren't so aware that a basic science advance could quickly be translated into a therapeutic modality of some kind. And we are very aware of that today. And therefore, it is a question that comes to your mind in this case, but would come to the minds of people in my lab. Actually, Luke is an MD PhD, and so he may well, in the future, think that way.
Pierre?
What percent of eukaryotic proteins are expressed from unspliced mRNA?
Sorry, let's have that again.
What percent of eukaryotic proteins
Oh,
are expressed from unspliced mRNA compared to the retrovirus?
yeah, there are only a few genes that give rise to protein without splicing, particularly histones. But there are others. I don't know what the number of such genes is, but it's small. Yeah.
How do these bottleneck introns affect alternative splicing?
Now there's a very interesting question. He asked how the bottleneck introns affect alternative splicing. I haven't talked about alternative splicing. And interestingly, in the inflammatory response, the genes that are activated, very few of them have alternative splicing. So this is not a good system to even ask that question. But I was surprised when we did ask that question and discovered that it wasn't really relevant to most of what we were doing. Because there are, I mean, if you look at random genes, there's a lot of alternative splicing. Yeah. So she points out that the control of BUD13 becomes an interesting question. From what we've been able to do, we don't see much control of BUD13. It's pretty well a constitutive gene product. And there are many housekeeping genes which are sensitive to BUD13. So it's not particular to inflammatory genes, for instance. And so I think it's a constitutive mechanism for dealing with these kinds of introns, which may not be under complex control. Now, you asked what happens if we overexpress it. You can increase the amount of splicing by artificial overexpression.
Maureen?
My question was related to her feedback because, for example, in HIV infection, we actually see crystallized interferon in cells in kidney, as well as in lupus.
Yeah.
And so is BUD13, can that be altered by viral infection or, say, inflammatory responses in lupus, where you also see
Yeah.
these infusion bodies, which is appropriate for a Palade lecture since we're electron microscopy.
It would be I mean, she asks whether I know anything about whether it's involved in interferon overexpression, and I don't. I just don't. The question in relation to HIV is an interesting one, and we used to do a lot of work on HIV, but that hasn't overlapped with the guys doing this work. Well, Bud13 is part of a complex of proteins that are bound, that are spliceosome proteins that bind to intron sequences and that are involved in the biochemistry of splicing. Whether there's a helicase in there or not, I don't know. Bud13 is not a helicase. I'm losing you, yep.
Yeah, I said before, perhaps the new mechanism described just actually introduce another layer of kind of fine tuning of the disease. So the idea is that because many of the genes, they have multiple level of integration, therefore, in the splicing, so the further you lost this one, so maybe just the dynamics, the derivation is avoided, but in fact, in the weak people found the system is more noisy, you always have evolution of the
Yeah.
My question is, whether or not the bioprotein, after high specificity, that could change its association with the physical location of the origin of the cell and the fulfill the
Yeah, that would be a very interesting experiment to do. With the technology available today, you could actually identify where in the cell Bud13 is. And I think that would be interesting to know. I agree with you, it is certainly part of a multi-layered regulatory system. Just we can't really put it in its place yet.
One final question, way up the back.
What you're really talking about is alternative splicing. And alternative splicing does give rise to multiple proteins from the same transcript. But in the case of what we're talking about here, we don't see that. Most of the introns that are left in there are not translatable. They have nonsense codons in them and throw you out of frame, reading frame. So they're not actually, at least the ones we've looked at, are not leading to alternative proteins.
I'd like to thank Dr. Baltimore for his inspiring presentation.
Thank you.